Thanks so much for the invitation to give an update on where we're going with the ELITE study. I'm here on behalf of several of my colleagues, so we'll see them at the end. Let's see. There's been posited several determinants of what allows elite level or human athletic performance, and so these are some of them. On the left, you have some of the intrinsic factors, including sex, height, VO2 max, and skeletal muscle traits, and then there are a number of other traits that have been proposed or been associated with elite athletic performance. There's also obviously extrinsic factors such as coaching and desire and socioeconomic opportunities. All of these go into what takes someone from the potential to have elite athletic performance to actually being a world or a national champion. There have been several studies over the past now more than 50 years looking at heritability of some of the factors that go into athletic performance. Chief among them, this measure of VO2 max. And so as you can see here, these heritability estimates, largely from twin and family studies, show that VO2 max is significantly heritable to 40 or 50%. So the absolute max that one might achieve in one's lifetime estimated to be about 40% heritable, and then the ability to be trained to a high VO2 max, as shown in this study by some of our collaborators, Dr. Bouchard and colleagues, shows that the trainability is almost 50% heritable. And so we took these as a sort of motivation to then look at how we might better explain some of this heritability. So what is VO2 max? Well, it's a maximal oxygen uptake that one can achieve through one's body during exertion. And so on the left is, the top left is the current world champion in VO2 max, who's a young cross-country skier and biathlete. On the right is one of the most famous patients with one of the highest VO2 maxes ever, and we'll talk about him later. This is him winning the Squaw Valley Olympics in the 4x10 cross-country relay. This is a gentleman testing in our lab wearing a mask that's allowing measurement of his oxygen exchange and carbon dioxide output. And then this is the oxygen uptake curve from yours truly doing an exercise test after about 10 years of detraining related to cardiology fellowship, training, instructorship, et cetera. So there's a range of genetic association that's already been described with VO2 max, and so the gentleman who was champion in Squaw Valley is actually the first convicted genetic doper in the world as far as we know. So he actually had a variant in his erythropoietin receptor gene that truncated it and caused it to be constitutively active. And this was dominantly inherited through his family of multiple world champion skiers and biathletes. And so this is a picture of him winning one of his many illicitly attained gold medals. Of course, he was born with this, and so he is ultimately freed from his previous doping allegations because he had a very high hematocrit as a result of this. This didn't free him from his admitted use of amphetamines, which were illegal performance enhancers. And on the right, again, is our colleagues, Dr. Bouchard et al. In Louisiana, doing really the only genome-wide association study to date, looking at common variants associated with trainability. And so both of these studies show sort of different mechanisms one might get to high performance. And so there's a recent study in, there's actually a popular article that you might have heard of from This American Life. ProPublica and This American Life did sort of an article in collaboration on these two individuals. They both have mutations in a gene that actually we've recently completed a clinical study in patients with LMNA mutations actually can have cardiomyopathy, progeria, or lipodystrophy. And so both of these patients have lipodystrophy, but one has muscular dystrophy, and one has sort of super muscle strength. And so the woman on the left is a Canadian Olympic medalist. On the right is a woman who is wheelchair-bound because of her muscular dystrophy. And they have mutations that are 3 base pairs away in the same gene. And so this is a pretty amazing story, and I encourage you to go to ProPublica, or you can look at my Twitter feed, and I have a link there. So we have a goal with the ELITE study to discover genetic determinants of physical fitness in the world's most elite endurance athletes, but we also want to use this to provide an impetus for finding additional genes that may be associated with various types of disease. So this is, again, going back to VO2 max. So this is data from this Swedish study looking at literally thousands of women and men and looking at the normal distribution of sort of a lifetime or best VO2 max is. And so we're looking in the ELITE study at the far right. So people who are better than 1 in 7,000 in the population based on Swedish data, and probably better than 1 in 100,000 in the population if you include all humans, because there seems to be some advantages in the Swedish population that isn't present elsewhere in the world. So we've spent a lot of time over the past few years, I believe it's 4 years that we've been recruiting cohorts. Athletes have been recruited from around the world. We collect basically the data from their lifetime best VO2 max, as well as a DNA sample. They fill out a consent form, of course, and a survey about their performance. And today we have samples from more than 650 athletes on site in the lab. And we've now sequenced 263 on enhanced exomes. I'll show you some of that data in a minute. And we've had 500 whole genome-wide SNP chips run. Those data were supposed to be here last week, but they've been delayed a few weeks, so we'll be hearing about that soon. And then with future collaborations, as shown in the blue dots, we have access to an estimated group of about 2,000 super athletes or super endurance athletes that we should be doing the genome-wide study on over the next few years. So these are all of our collaboration sites, led by the one in Sweden here, and that's Mikael Mattsson, who's been a visiting scholar with us for a couple of years now and is really driving the recruitment of this project. So how do we find genetic loci that may be responsible for some component of our participants' VO2 max? So there's really 3 techniques that we're looking at. One is to do small family studies, so we do have some SIB pairs and father-son or father-daughter combinations. But really, we're in the modern era of hypothesis-free genetic testing, where we're looking at genome-wide association studies and whole exome sequencing in this case to identify loci that may be associated with VO2 max. So the first set of samples we've done enhanced exome sequencing, and in part helped by C grant we got from the Wisdom Group. And so they've been sequenced on the HiSeq 2500, that's supposed to say at Personalis, which is a local company here. The data is highly filled in, so we have 8,000 genes where there's at least 99% coverage of bases, whereas other exome technologies may have about 95% coverage. And the cost was quite reasonable as this is on a research basis. So these are the distribution of VO2 maxes, or maxi, for our participants in the exome study. And so it looks like these the women and men are separated by some force of statistics, but actually these are the best that we've gotten. So there's a natural separation between the best women in the world and the best men in the world. and that's no selection was done on our part to try to enhance that, except for we got so many participants in the 75 category that we didn't need to go lower than that in men. So we looked at a portion of our exome data and thought we'd start at the sort of easy place, which is genes which have new stopgain variants, so variants that would be expected to interfere with the normal production of protein, or at least may contribute, if in a dominant negative fashion, to a change in that protein. If you look at some of our colleagues in the athlete or sports genetics world, there's been over 530 genes that have been proposed to be associated with some component of physical fitness, and so we looked in those, and this is a list of those genes that have stopgain variants. We have our own list of cardiac candidate genes, being cardiomyopathy lab, and so there's also some stopgain variants in those genes. And then we also have a list of mitochondrial genes. We might think that having some enhanced mitochondria might benefit you when you're trying to win the Olympic distance triathlon, as this woman from Sweden is doing. Then we also have stopgains in those. If you look at all 20,000 genes, the list is so small that it runs off the slide. We also looked in the same data set across a gene and said, well, if we're looking for rare variants and we have only a small number of samples, can we find genes that have more rare variants than we might expect? And so this is known in the genetics literature as burden testing. And so we rank normalize the VO2 max, and then we look for excess of rare variants. And we see several genes that are not genome-wide significant yet, but this is only a subset of our data. And so we're hoping that with a little bit more data, we'll reach genome-wide significance and find genes that may have a clear excess of rare variants. Within those genes, we did find differences comparing our male and female cohorts, and so we found functional rare variants in some highly plausible genes, including the androgen receptor. So you can imagine if you're a woman who has an abnormality on your androgen receptor, that you might have some advantage in terms of either muscle bulk or lack of muscle bulk, both of which could help with certain types of exercise, as well as FTO, which again is helps with body composition and energy management and energy utilization and efficiency. So these are plausible disease gene candidates, but obviously more work needs to be done on these specific variants to determine if they're truly causative or spurious. We also, with one of our new talented graduate students in the lab, started looking into copy number variants and found copy number variants that are overrepresented in several mitochondrial genes. And so, again, further work will be needed to determine if these are affecting or are significant. And so at least one of these is a very common copy number variant seen in about half of our samples, either as a deletion or a duplication. So it'll be interesting to see if we can see this in some tissue samples that we have available to us. We also have a cardiomyopathy cohort, again, working as a cardiomyopathologist. We have a cohort of about 48 patients who've undergone a whole genome sequencing, and so we sort of create a digital exome from that, and we can compare what we find in those digital exomes to the elite exomes. Again, with the subset of the data currently available, we were able to do rare variant burden testing in both cohorts and found only one gene that had that was significant at nominal p less than 0.05 in both samples. This is a very large gene, the cardiac renin receptor, and we were well aware of its association with different phenotypes in cardiomyopathy, but had actually found recently some new data suggesting that it's also important for exercise capacity, so that's a lead we're going to follow up on. And then, unfortunately for today's talk, we have not gotten the full data from our genome-wide association study, but we do have a pilot genome-wide association study of 3,500 total samples, 3,000 controls, and 500 from our pilot phase of the ELITE study. This is a chip that has been specifically designed for multi-ethnic genotyping, so we can do this for our worldwide cohort, and it has 1.7 million SNPs on it. And so the data, again, is hopefully coming within the next few weeks, and then we'll do our full analysis from that. But we're able to use our exome data to do what we call a exomic GWAS, looking only at variants that are within the protein coding region. And if we do this either by the absolute number of VO2 that doesn't correct for body size, or if we do for VO2 max corrected for body size, then we have two different variants and two different genes that reach nominal significance. And so we're interested to follow those up. Interestingly, both of them also have disease-related phenotypes that have been previously described, but very sparse data from that. And then, again, from a preliminary standpoint, we did do sex-specific VO2 max, so this is trying to say what proportion of VO2 max is different between sexes, which is really the goal of our project from a sort of sex differences perspective. Unfortunately, we don't have sufficient numbers to generate a significant p-value, but we're hopeful that with our larger sample that we will find some sex-specific differences, and then we can start working on those to figure out why. So with that, I'd like to thank our Stanford investigators, you and Ashley, who's currently in the UK, Mikhail, who's currently in Sweden, Daryl, who's in the back of the room, who's done a lot of the data analysis with us, and Alex, who's busy coordinating studies back in the lab. So there's a lot of other people who've been involved that are mostly spread around the world, and hopefully as we move forward, we'll bring some of them in here to teach you more and teach us more about what we're learning.